Thank you for the organizers for having me come and speak about the rapid analysis of drug resistance and tuberculosis in our evaluation. So we are from Mayo Clinic. We have sites all over the world, but our three main locations are actually in the United States. We have one in Jacksonville, Florida, one in Scottsdale, Arizona, and the original and largest site is in Rochester, Minnesota, where I am from. We have more than 76,000 employees and a staggering 1.4 million unique patients that we see every year from 130 different countries. I have been supporting the Mycobacteriology laboratory as a developer under the direction of Nancy Wengenek for 22 years now. The lab is staffed 24/7, 365 days of the year, and our lab has grown quite a bit in the last few years. We now have 56 employees. The organisms that we manipulate require that we have to have respiratory protection, along with engineering controls, such as biological safety cabinets and BSL-3 rooms. And in our last renovation, we now have 9 in our laboratory. We perform over 48,000 mycobacterial cultures annually. And we are a CDC-regulated or designated U.S. regional and training and medical consultation center for tuberculosis. So some of our test menu highlights, as I stated, we do a lot of culturing, but we do acid-fast staining, we do antimicrobial susceptible testing, and we perform approximately 5,000 phenotypic susceptibility tests annually. We have an extensive molecular menu that includes Sanger sequencing, next-generation sequencing, and real-time PCR. So Sean, I'm going to ask, why is M. tuberculosis so important? Well, it's highly transmissible bacterial pathogen and is the causative agent of tuberculosis. And it still kills 1.3 million folks per year. What's more alarming is that a lot of multi-drug resistance MTBC strains have emerged, and this is reducing the viable treatment options, and therapeutic inventions are becoming more costly and time-consuming. The current methods that are available for detection of M. tuberculosis drug resistance require weeks to months, and this delays the administration of appropriate therapy. And you can't hide from TB. It is everywhere. We see it in the news headlines often, in schools, and traveling, all around the world. This slide demonstrates our turnaround times for some of the tests that we offer in our laboratory. We're actually able to analyze by real-time PCR directly from clinical specimens and do AFB smears in 24 hours or less. The bulk of our work, however, comes from solid and liquid culturing, which can take anywhere from 2 to 3 days up to 42 days. And once we grow that organism, we're then able to identify it either by MALDI-TOF mass spectrometry, 16S ribosomal DNA sequencing, and/or real-time PCR. Once we get that result is then we can do our phenotypic susceptibility testing, which can take anywhere up to 60 days. So this is where the gap is and where we hope Oxford in the future could help with us, the clinical aspect. So we are looking for a rapid and less labor-intensive method for molecular detection for drug resistance. And as I stated before, the phenotypic susceptibility takes weeks and delays an inappropriate treatment. And our current methods that we have evaluated either are limited by just the targets or they're difficult to implement and sustain in the clinical laboratory. So we evaluated the TBDR assay. And it's great because it enables a simple and rapid analysis of Mycobacterium tuberculosis complex for drug resistance. What's great about it is that it amplifies 24 different genes found in Mycobacterium tuberculosis, and it sequences them to identify specific SNPs which could confer antimicrobial resistance. In addition, it utilizes the HSP65 gene, which can identify other mycobacteria, which could also be important in the future. The workflow is pretty slick. It amplifies the target genes present, it does rapid barcoding, and then does the real-time nanopore sequencing on the GridION. So, how did we evaluate the TBDR assay? Well, we looked at 2 different specimen types. We looked at culture isolates, And we also wanted to know how this assay would perform on respiratory specimens, because we often have issues with sensitivity from respiratory specimens from a lot of our genetic assays. So once these specimens were processed according to our standard protocol, the DNA was extracted and then run through the dr kit. 22 samples, which is a pretty good size for this kit, which is really nice for us as well, were processed. Again, the three main steps that included the PCR, the rapid barcoding, and the pooling of samples. We then loaded it onto the GridION. The real-time data was collected through the Minnow software, and then that was uploaded into Epitome, where it was analyzed. And any SNP that was detected was compared to the WHO catalog of mutations for M. tuberculosis. And before I go back, there is, on that table there, we've listed the 24 genes that were detected with the assay and then associated with the drug class. So pretty extensive. We were excited about this. So how did it do? Well, actually really well. Here are the results directly from culture isolates. In this table, I listed the number of variants that the Oxford Nanopore TBDR assay detected for each of those drugs. And then we wanted to compare it or correlate it to the phenotypic susceptibility testing that we did within our lab. We did a % agreement, and then if we had any discordance that we were able to resolve, we then did a corrected agreement. So overall, rifampin, the fluoroquinolones, which include moxifloxacin, ofafloxacin, and streptomycin, amikacin, and kanamycin, did fantastic right off the bat. We had 100% agreement between phenotypic susceptibilities and the TBDR kit. Now, isoniazid, ethambutol, and parazinamide, actually, Oxford picked up more variants than we were able to detect with our phenotypic susceptibilities. And upon further looking at them further, we were actually able to resolve those discrepancies with a third next-generation sequencing assay, confirming that ONP TBDR kit was actually correct. And we had the correct agreement there, so it looked fantastic. So then how did it do for respiratory specimens? Also fantastic. We compared it, again, the TBDR direct from specimen, and we compared it to how we identify our respiratory specimens post mycobacterial culture. And we were actually able to pick up 43 of the TB specimens that we tested. And then the 63 were actually non-tuberculosis mycobacteria that were also identified by culture, and some of them by the Nanopore. So again, another niche that maybe Oxford can fill for us. So overall, we were super excited. The workflow is really rapid and simplified to a lot of the things that we evaluated in the past. I know somebody had asked earlier about the time that it takes to do this. We could do this in under 8 hours, from extraction to answer. The TBDR assay performed well on both culture isolates and respiratory specimens. And we did good agreement with the TBDR assay with our phenotypic susceptibility testing. And where discordants were found, we were actually able to resolve them in favor of the Oxford Nanopore TBDR assay. And this suggests that the TBDR assay, once approved or offered, would offer a good clinical testing gap that we are now missing in our laboratory. And that is all I have.